In the shadowy corners of the microbial world, few agents of disease inspire the same primal fear as rabies. The perpetrator is the Rhabdovirus, a bullet-shaped particle engineered by evolution for a single, terrifying purpose: a relentless and fatal assault on the nervous system. This ancient scourge has plagued mammals for millennia, and its reputation is well earned. Once the overt signs of its presence begin to manifest in a host, the outcome is almost invariably death. The question that has haunted scientists and physicians for centuries is, how does this microscopic assassin carry out its deadly mission with such unfailing precision? It is a journey that begins with a single breach and ends with the complete capitulation of the body's central command. The devastating odyssey of the rabies virus begins not in the brain, but at the cellular level, often in the humble muscle tissue surrounding a bite wound. Upon entering the body, the virus doesn't immediately storm the nervous system. Instead, it executes a more insidious strategy. It latches onto specific receptors on the surface of a host cell, tricking it into granting entry. Once inside, the virus sheds its protective outer coat, releasing its RNA genome into the cell's cytoplasm. In a masterful act of biological piracy, this genetic material hijacks the cell's own reproductive machinery. The host cell, now a puppet, is forced to abandon its normal functions and dedicate all its resources to producing thousands of new viral components. These components self-assemble into complete, infectious viral particles, which then bud from the cell membrane, ready to propagate the infection and begin the next stage of their sinister journey. Having successfully multiplied at the site of entry, the viral army launches its main offensive. The newly minted viruses seek out and infect nearby nerve endings, the peripheral communication lines of the nervous system. This is the critical turning point in the disease's progression. The virus embarks on a slow, methodical, and unidirectional journey, traveling retrograde along the nerve axons. It moves relentlessly, centimeter by centimeter, towards the spinal cord and, ultimately, its final destination: the brain. This journey can take weeks or even months, a silent incubation period where the host remains unaware of the impending doom. Once it breaches the central nervous system, the virus unleashes chaos, spreading rapidly throughout the brain and triggering severe inflammation or encephalitis. It colonizes key areas like the hippocampus, brainstem, and cerebellum, leading to the catastrophic neurological symptoms that define the disease. Transmission of this lethal agent is most commonly facilitated by the saliva of an infected animal, usually delivered through a bite that breaks the skin. While rabid dogs remain the principal source of human deaths globally, a host of other mammals serve as reservoirs for the virus. Bats, often carrying the virus without showing symptoms themselves, are a significant source of human cases in the Americas, while raccoons, skunks, and foxes are also common vectors. The initial symptoms in humans are deceptive and easily mistaken for a common flu, including fever, headache, and general weakness. However, this prodromal phase quickly gives way to the terrifying and specific neurological signs of clinical rabies. The patient may experience severe anxiety, agitation, confusion, paralysis, and hallucinations. The most infamous symptom is hydrophobia, a horrifying and painful fear of water triggered by involuntary spasms of the throat muscles when attempting to swallow. Diagnosing rabies in a living person presents a significant challenge, as symptoms can be nonspecific in the early stages. Clinicians may test samples of saliva, spinal fluid, or a skin biopsy taken from the neck, using techniques like the fluorescent antibody test to detect the presence of the virus or its antigens. Tragically, by the time a diagnosis can be confirmed through the presentation of classic clinical symptoms, the window for effective intervention has long since closed. There is no cure for clinical rabies. Once the virus has established itself in the central nervous system and symptoms appear, treatment is purely supportive. Medical staff can only manage the agonizing symptoms and provide comfort care as the disease runs its inexorable and fatal course, typically ending within a matter of days. Yet, despite this grim prognosis, there is a powerful and effective defense against rabies, and it lies entirely in the realm of prevention. The moments immediately following a potential exposure are absolutely critical. If an individual is bitten or scratched by an animal suspected of being rabid, the first and most important step is to immediately and thoroughly wash the wound with soap and water for at least 15 minutes. This simple act can significantly reduce the viral load at the site of infection. This must be followed by a visit to a healthcare provider to receive post-exposure prophylaxis, or PEP. This regimen, which includes a dose of human rabies immune globulin and a series of rabies vaccine injections, is nearly 100% effective at preventing the disease if administered promptly and correctly. On a broader scale, the cornerstones of keeping our communities safe are the vaccination of domestic animals, especially dogs, and maintaining a respectful distance from wildlife. Through vigilance and prevention, we hold the power to stop this incurable virus before its deadly journey can even begin.